So we got calls from haplotype caller, and as I mentioned before, haplotype caller tries to output as many variants, even if they're not the best quality, with the reasoning that we will filter them out later, the things that we don't want. So there's a few approaches we can take. So we'd like to get rid of things that are potential false positives, and we have access to information such as annotation of those variants. And if, for example, we see that a variant is some kind of outlier with respect to those annotations, we might filter it out based on that. So that is one of the kind of first filtering approaches that was done, is to just set a hard filter on some annotation, and if, for example, like a mapping quality, and if the mapping quality is less than a certain value, you just get rid of that variant altogether. The next in evolution is to have a more sophisticated machine learning model, and that requires to have truth resources. So we need some kind of validated call set where we know which variants are true, and also which variants are false, which we will discuss how we figure that out later. And the latest and the greatest in evolution of the filtering tools is the ones that use some sort of neural network technology, and that also requires to have truth resource. But this now takes, instead of just using the annotation information as the first two approaches have done, we now can manage to also look at the sequencing context of that particular variant that we're looking at, and in addition to annotation, and we can also take advantage of the reads and so on. So that's sort of the story of the variant filtering in GATK. So the hard filtering was, it's okay for some purposes. The problem is, however, that, well, first of all, it's not super accurate, because you just basically cut the space of your variants cut the annotation space in half with this kind of rough way, and you're not really you don't have ability to define some kind of nice smooth boundaries in the space. So you might get rid of a lot of true positives as well when you do that kind of thing. And the second problem is that this is very specific to current call set, and then once you get another call set, you have to adjust those boundaries, and there's no way to automate it. So the first kind of attempt to automating it is by using the more standard machine learning approaches, such as SVM, random forests, and the decision that is implemented in GATK is in VQSR uses Gaussian mixture model. And here we we also take advantage of the same annotations, but now we can automatically learn it given the new call set. We don't have to manually adjust those decision boundaries. We can just run our algorithm and we get results out without any manual intervention. However, that doesn't take advantage of any sequencing information at all. We're just looking at a few covariates that that particular variant has, but we can also take advantage of the sequencing context, and that's what the more modern approach also implemented GTK does, which given we implement the architecture of the convolutional neural network, and we provide it with the sequencing context and the annotation, and we have our truth data, and we train a neural network, and we do filtering that way. Okay, so a few words about the annotations. So the annotations I'm talking about are basically output in the VCF in the info field, and those annotations are, like, for example, you get the allele frequency, you get the depth of coverage, you get mapping quality, position rank sum, I believe, is one of them. It's not mentioned here, but basically, usually VQSR takes advantage of about 7 of those annotations, and those, well, you can see how some of them can actually give you an idea about how confident you should be in that variant, right? Such as like allele frequency or mapping quality, depth of coverage. The lower the depth of coverage, the less sure you're about that variant, and so you want to train your model to be able to take advantage of that.
So is the VCF file?
Yes. The final VCF that is output by.
So one VCF file that is after joint calling.
Yeah. After this is done after the genotype refinement stage. So this is what annotation space looks like when we project it down to these two specific dimensions with 2 annotations that are not important for this particular reason, but the point here is that, well, you see that we're trying to do some hard filtering here. However, well, in this case, we don't get rid of all of the false positives. Ideally, we'd like to keep all these kind of big, nice blob of variance here in this annotation space. But however, we can really choose the thresholds to perfectly align with the edges of this blob. Right, and this is a picture once we train the model. Once we train VQSR, again, the red blob is the good stuff that we want to keep, that passes the filter. The green stuff is the stuff that the variants that are not going to pass the filter, but they're not as bad as the blue stuff, and the blue stuff is not as bad as the purple stuff. So each point will be assigned this VQ slot score, and that's what the colors represent. Let's see what else. Okay, so how do we get truth data? So the idea So the idea in that people came up with in the truth resources that currently exist, such as Genomics in a Bottle, and so on, is to have a call set that is a consensus call set with many different callers. And if you get a bunch of different callers, such as GTK, Freebase, the Illumina ones, and they all agree on the same call, then it's probably a good call. And so we put it in this confident region, right? And so we have a validated VCF which has all the confirmed calls, and now once we get an unvalidated VCF that we would like to validate, if the call agrees with the one that is in the confidence region, we say it's a true positive. If it does not, it's a negative, right? Another more clever way to make truth data that was done recently is by basically taking these hydatidiform moles, which are basically haploid. These are haploid human cells. And we use PAC-by sequencing to sequence this haploid DNA. So we take two cell lines, we sequence them separately using PAC-by, which is an orthogonal data to Illumina, and then we map it and we mix it in silico. And now what we can do is we can actually mix it in a test in a tube, and we can sequence it with Illumina, and this is the call set we try to validate. And so because these are haploid, they're actually they're easy to call. And even though we're using PAC-by, which has long reads and it has a problem of having a lot of base errors, it's still the fact that it's haploid makes it easier to call and makes it a good truth resource. So there's a lot of ways to make a good truth resource, and so you want to take advantage of that fact to be able to train a nice machine learning model. So here's the VQSR in a nutshell. The basic idea here is that we have a bunch of variants that we believe, and we train our we fit the variants that we believe with a few Gaussians. So we fit a mixture of Gaussians to it, and we have, say we have like, we say there's like 5 Gaussians. So we fit 5 Gaussians to this data set to only good variants, and then after we've done that, we look at everything that's outside of those 5 Gaussians, and we say, well, those are bad variants, and we fit another Gaussian mixtures of those. So now we have two models. We have kind of the good variants, the model for the good variants, and the model for the bad variants. And now, given the new variant, if you want to assign a score to it of how good we believe it is, the VQ slot score, we just take the ratio of the good model to the bad model. Right, so if the point is like anywhere here, where there's going to be a high probability of this variant being in the bad model and low probability being in a good model, we assign a low VQ slot score. And so that will be one of those variants that will end up somewhere here in one of the bad trenches. Right, and so now that we assign the VQ slot score, we can filter the variance given the threshold of the VQ slot that we will just throw out of our call set. So this is done per per variant in the VCF. Yeah. Any questions so far?
So basically, you are not recommending the hard filter.
Yeah, we don't recommend hard filtering now. I mean, it depends what kind of resources you have, but if you can do it the other way, then definitely. And the current machine learning models that we have are actually pre-trained, so you don't need any resources to be able to run them. Okay, so that was that was the QSR, which only used annotations to filter the variance. But what about all the other stuff that we don't include in the information that our machine learning model uses? So one thing to look at is the sequence context, and this is a Lego plot. Have you guys have anyone not seen it before? So basically, the idea is that if you just like look at this little square right here, this indicates the sequence, the two bases, left and right, to the current base. And here in the green, you can see this represents A to G SNP. So this is basically a distribution of A to G SNPs given the surrounding bases. And this is done for different all the different SNPs. So the point here is that the sequence context matters in terms of how we call SNPs. And so this could be due to actual biological reasons for why SNPs occur more with this context, or this could also be due to the fact that it's harder to call things with those, harder to call or harder to sequence things with this particular context. So for example, for example, this big yellow bar is a C to T SNP, and the in the region where in the context where the surrounding base is a T, so it's a poly T region, which is harder to call. So the point is that it would be nice to take advantage of this information in order to do variant filtering. So yeah.
Just maybe silly question. Did anyone try to like compare different organisms, how it looks for different organisms? Then you could rule out the sequencing issue. Maybe the biological things.
I see. Yes. So yeah, you could see how much of it comes from actual biology by doing that, but I have no idea.
Okay.
That would be interesting.
And this kind of soft thresholding applies only for the SNPs, not for any INDELs.
I'm sure similar things happen for INDELs, but this diagram is only for SNPs. So, okay, now we have the motivation to why we want to look at the sequence context. Now let's find a way to kind of to represent it so that we can take advantage of all the convolutional neural network research to apply it. And the one of the most natural way to represent it is using one-hot encoding. So every base will be represented using these 4 channels, A, C, G, T channels. T base will be just a bunch of zeros and 1 in a T channel. A base will be a bunch of zeros and 1 in A channel. And we can as well represent absence of the reference information. So we can just represent N as 1/4 in all of the channels, right? So we can also represent uncertainty in this way. Okay, so this is the way we can represent numerically a reference as the tensor, as this matrix that we will eventually put our neural networks. How many people actually have seen or used neural networks for anything? Cool. So, I mean, one thing is they used for very well for image processing, and the convolutional neural networks that I use for image processing, they take advantage. They look at the features of the image, and they have a lot of layers. So as we progress towards deeper layers, they look at more and more complicated features. So the idea basically is that we're trying to do the same thing with the reference. You want to start by looking at the very simple features, and as we get deeper and deeper in neural network, we'll get more complicated features out. And I'll show you examples of those features. Okay, so now we have the reference represented, and now we want to do something with that reference. And what we do is we apply a position weight matrix. So for example, consider this example of multiple sequence context, which was where we have multiple sequences, and we're trying to kind of get the multiple sequence alignment here, and we see that these G's and T's here are the most important bases in this multiple sequence alignment. And the way it's represented is in this position weight matrix, you have higher weights in these particular positions, right? And the other stuff is not important, so these other numbers are much lower and much less important. And so what this position weight matrix encodes basically is that it encodes a motif that where these G and T right here are very important, and everything else is basically we don't even really care about, right? So this is sort of a motif finder, the way to represent a motif finder. And the way we now can use it, so now we have a sequence, and now we have our position weight matrix, and what we can do is we can go along this sequence, and we can apply this position weight matrix. We can convolve the position weight matrix with a slice of this sequence, and if the sequence has the motif that is represented in the position weight matrix, we'll have our neural network will light up, and we'll be happy. And so we can convolve the entire thing and get some output. So the basic idea is that that's how we can encode our reference and apply the neural network convolutions to that.
So motif is derived from the samples, and that the sequence is the reference sequence.
I'm sorry?
The motif is derived from the samples, and the sequence is the reference sequence.
Yeah, the sequence, the only input we give is the reference sequence. So the input will be the reference, the window of the reference sequence where the variant is, and the output is whether this variant is true or false. There's no we don't input reads or anything else, just the sequence information so far.
Position matrix, is that obtained from the alignment of the reads to the
I'm sorry?
The position matrix, is it obtained from alignment of reads to the reference?
Position matrix we learn. So I will give you an example. I think maybe it'd be easier to start with this. Basically, the position weight matrix in the beginning is initialized to something very random. And then as we train our neural network, so we have our inputs, which is the reference, and then we have these convolution layers, which are represented by these position matrix position weight matrices that we're trying to learn. And then the output is whether this variant is true or false. And then we do our deep neural network magic, the backpropagation, to try to learn the weight of those position weight matrices. And this is like what happened. Yay. Sorry about that. Yeah, okay. So this is an example of our motif finder in training. We start off, these are completely random weights, just drawn uniformly. And yeah. Oh, so we just draw like weights from uniform distribution. So like each of this is drawn from uniform distribution, and we crank our deep learning machinery and train this, and finally we get this nice poly-C motif finder. Right, so this is example of some of the motifs that the architecture, the training found, and this is kind of reaffirming because we found the motifs that we knew previously to be important with respect to how hard it is to find variants and how sure we are that those variants are real in these regions. So, for example, this is a motif that finds an STR regions with the CT repeat, poly-C motif that we saw previously, and then there's this thymine edge detector when you have a bunch of T's, and then we don't have any T's. So these are the positive ways, and this is the negative ways to represent that we do not have them there.
How do we decide the window of the motif?
Sorry?
How do we decide the window of the motif, or is it some window? How long window will be?
How did we decide that?
Is it some kind of dynamic parameter that stretches
the window itself? Yeah, it's just an input parameter. So that is encoded in the architecture. The size of the convolution filters is encoded in the architecture. Yeah. So that's kind of predetermined. How like it was chosen, I'm not sure. Partially, a lot of the hyperparameters in the architecture was varied and then validated, and we saw how well it corresponds to how well we perform with each particular parameter selected. And so we train a lot of these position weight matrices at once. Simultaneously, and yeah, that's the idea. So that's our one-dimensional CNN model that only takes advantage of the sequencing context around the variant. Now the next step is to add the read information to the reference. So basically, we want to encode this IGV screenshot that's located around our variant of interest and put it in the tensor. So before we had just like one hot encoded reference with ones and zeros. Now we're gonna have a three-dimensional thing that encodes all of the different things about the reads around our variant, such as the bases themselves, the whether it's in reverse strand or forward strand, whether it's first in pair, the mapping qualities, the base qualities, and so on. So all of that will be somehow encoded in our neural network. And so this is just to give you an idea. That is what the input will look like. This kind of this block that represents everything in the BAM around this particular variant, the entire context. So, for example, let's see. These guys in front are the reads themselves. Then there's the reference behind. Then we encode the information about the reverse strand, or whether the mate is a reverse strand, with more channels. So basically, these are in this dimension we have the channels of our tensor, and in this dimension we have the reads themselves. And this is in this representation. This is what the variant look like. So this is in the more brighter colors we have reads. In the more transparent colors behind we have the reference, and this is a little snip right here, right? This is a G2A snip. So we have half of the, so the reference is G, and we have half of the reads being RA. And we can also represent indels in a similar manner. So yeah, we can represent all kinds of variants in this tensor representation. And so this is, on the right is the example of natural image processing. And there's a way to show what kind of patterns excite neural networks. And in the case of natural language processor, this is the very first layer, and then as we go to the right, to the second, third, fourth, and fifth, and as we go deeper, we get more and more complex feature out of our images. So we start with like very simple edge detectors, and then there's some patterns, and then you actually start seeing some faces, or yeah, some more complicated features. Nose, I don't know. And the idea is the same for our convolutional neural network for variant filtration, as we start with very, very, very rough feature finders that kind of say, oh, whether it's whether we have a head side or not. And then once we get deeper and deeper, we get into more granular kind of granular shapes that look at specific context around our variant. This is the architecture. This is this is the architecture for the 2D CNN model. So we start with, so this is the input, the tensor input that represents the reads and the varied context, everything we talked about just now. And then we have a bunch of convolutional max pooling layers, not really important. And then we get to a point where we also take the annotations, the same annotations we used for VQSR, and we just append it to the output of this part of the network. And then we have some some dense layers, and finally we output whether it's a real variant or whether it's an artifact, right? And so then we train that network, and we get results. So these are results for different different samples and different tools. And on the left you have indels and right SNPs, and we outperformed the VQ star, and we also outperformed the deep variant, which is also the a variant filtering. Well, it's both variant finder and variant filtering tool that Google made. And this is the value. This is the value under the the precision recall curve. So it kind of gives you an idea about not just one sensitivity precision, but overall the whole spectrum of them. Yeah. Different training sets, I believe. Different training sets, different. So all of them that were trained on one single sample, and these are three different samples, I believe. And like including also three different truths. Yeah.
So then asking the question, deterministic?
Is it deterministic? Yeah. So once you have the model trained, the actual applying that model is definitely deterministic. You just multiply a bunch of matrices. The training, hopefully, probably, I would say.
Assuming you're asking for the clinical application case, I think what you would do is train the model once and then systematically apply the same model. And as long as you're applying the same model, the results would be deterministic.
But it doesn't have different weights during the interpretation. So they are really fixed. They do not vary.
Yeah. Once the model is trained, all the weights are fixed. So applying is definitely deterministic.
So if you apply the same model to the same data over and over again, separately, right? To reproduce it, you will get the same results out of that.
Is it possible to cancel data at some point?
So I think that David Benjamin, the person who's working on MUTEC 2, that's sort of the next step, the natural step in the evolution of the MUTEC 2.
So they haven't validated it yet. Theoretically, it should be applicable, but there's some work to do to test how the tools actually perform and evaluate whether there's any need to tweak to adapt the tool for the somatic case, or whether it should be the boss. And that work has been started.
But you were mentioning the deep variant is not available for somatic samples. There has to be most likely the resolution for that.
Well, it's a different data type, so like you do have to retrain the model, and you have to possibly pick a different architecture.
Yeah, there's probably some adaptation that needs to be done. I can tell you, it's unlikely that deep variant will be adapted to support that, because the main two developers of deep variant are no longer working on that project. So it's unlikely that it will gain that functionality.
I mean,
however, the GATK CNN will that is being worked on. So that's a 3 to 6 months horizon. Can I say that? I'm just going to put some time on it, and then David will just have to make it happen. That's how it works.
I mean, yeah, if you would think about it as like in terms of looking at screenshot of IGV and trying to figure out a germline variant, I would say like most of the people would be able to say whether it's real or not. But with the somatic, it's like a different problem. So it requires a slightly more sophisticated solution.
Can I still ask about this? The training data is just one range in the sequence.
Yes, the input is just one context around the variant. So it could be like one chromosome, or it could be multiple samples. But yeah, what's shown here is just one single sample.
So I know that retraining the model is production from particularly for data and do you guys because there is a lot of new information.
Right. So what we did is, so this is actually described in this column. So yeah, we did an experiment of how generalizable the results are if we train on one data type. For example, like whether we train on one reference, or we train on exome, or we train on genome and colon exome, and so on, like all these different combinations, where the results are shown here. Right, so this is the difference in the RUC curves, and you see most of these values are okay. And what we actually use for production is we train a model using the combination of those. So it's kind of like, it kind of averages out, and we get a really good generalization across different data types. And so that's the model that actually would be used if you would just run GATK right now. Yeah?
Have you compared these to the deep SVR created by the Convolutions?
I haven't heard of that too. Deep SVR?
Yeah, it was published in Nature Genetics a couple of months ago.
Huh. I'll relay that, but no.
Second, have you actually tried random forest classifiers?
We did, yes.
And then sucked totally or something?
Yeah.
No, I thought they were okay.
I mean, oh gosh, we started looking at random forests 3 years ago.
We had an intern, I remember.
Yeah, there was an intern who started, and various people worked on that. And every time it was like, this is really promising, it's the future. And then something happened, and there were limitations that we kept running into. And at some point, somebody made the decision, like, this is not going anywhere, let's look at.
Yeah.
But yeah, that was totally explored, and it just did not pan out. I wouldn't say it sucked. I wouldn't go that far. There were too many limitations.
Yeah.
Yeah, I guess it makes sense because I mean,
Yeah, I think it beat, but like maybe not by much. So yeah, it wasn't pursued, and then we decided to go all in and train a deep convolutional neural network. Okay, this is basically the end of the talk. These are command lines. 1D model, 2D model. There's also there's also tools to actually train the models, which are not mentioned here. You can find them on the website, and these already have the weights of the pre-trained model stored somewhere. So you just apply it. And finally, once you apply it, so this only outputs the annotations. This just adds annotations to your VCF. And once those annotations are in your VCF, you define sort of the threshold above which you want to filter. And this tool, filter variant tranches, is what actually performs the filtering. Okay, this is it. This is it for the filter, variant filtering.